Good afternoon or good morning, I guess, depending on where you are. I am Khulud Jakaman. I'm an assistant professor at UT Southwestern, and it's my pleasure to introduce to you now our next speaker for this conference, Susanne Rafelski. So she's the Director of Assay Development at the Allen Institute for Cell Science, and I will leave it to her to tell you about all the cool stuff they are doing. But just to introduce her a little bit, before the Allen, she was an assistant professor at UC Irvine. And in terms of her background, her bachelor's is in biochemistry, molecular and cell biology, and math, all of them, from the University of Arizona. Then she did a PhD in biochemistry from Stanford University, followed by a postdoc at the Friday Harbor Labs and UCSF. And as you will see from her talk, and if you look at her papers, all throughout her career, her theme has been life cell imaging, computational analysis, image analysis, data analysis to get a quantitative understanding of the cell biological processes she is studying. So with that, I will take it to her.
Hi. I'm Susanne Rafelski from the Allen Institute of Cell Science, and I'm excited to tell you about our work trying to understand sources of variation in intracellular organization in stem cells. At the Allen Institute, we believe that we should be able to look at a cell and know what it is doing, what it did, and what it will do. And we believe that because there's such a strong relationship between cell organization and function. And if we know where all the parts, important parts that do important things in the cell are at a moment in time, then we should be able to know what the cell will do. The questions that we're interested in at the Institute are basic science questions: how to define and categorize cells and create a cell atlas. My favorite, which is what are the principles of cell organization? And then, of course, how cells change that organization as they move between states. The system that we work in is stem cells, and so we have a sort of differentiation and regeneration bent to what we're doing. And of course, we're very interested in how that applies to disease, especially in stem cells, which are such an important model system for human disease. The approach at the Institute is a holistic one. We care about the entire cell, all parts of the cell coming together. And our MO is really 3D life cell imaging because we care deeply about dynamics in our cells doing their normal thing. Our model system is the human-induced pluripotent stem cell, and its differentiation from this undifferentiated pluripotent state to the differentiated cardiomyocyte. We want to look at the most normal cells possible, and we want to look at structures in those cells. So we chose to do endogenous tagging with GFP using CRISPR-Cas9 gene editing, and then generate clonal cell lines with a huge amount of quality control, which we also release to the community. We also develop similarly well-established and robust differentiation protocols. Again, the microscope is at our center, and we do 3D life cell imaging of snapshots or time-lapse, and we add in ways of referencing our cells using dyes or other methods to be able to pull out individual cells out of a field of cells. We care about, again, the whole cell, so we have methods to work on integrating information from cell lines that only express one tag per cell line to get at structural organization in a holistic way. And we're starting to integrate other types of information, such as gene expression, in a spatial manner to integrate that with structural organization. Everything we do at the Institute is quantitative and has the purpose of being used for analysis and modeling, and also all of this together for distribution to our community for empowerment of all of us together. The model system that we use is, of course, the undifferentiated cell to the cardiomyocyte because it's a very well-established differentiation process, and because, of course, by the time you have the contractile apparatus, you've reorganized the cytoplasm tremendously. But we also have all of the beautiful cell states in between to start building a cell state space of cell organization in this particular process. Today, I'm going to focus entirely on the undifferentiated stem cell. But before I start that story, I just want to make one really important point, which is that if there's one thing I do today, if I get you guys to go to allencell.org and look at our website, the Allen Cell Explorer, so that you can see what we're up to and what we have available to you up there, then I've succeeded, and it's definitely worth a look. So we can take our cell lines one structure at a time, and we can image them and characterize these cells. And then we can also add positional referencing markers so that we can image these on a scalable integration assay for pipeline imaging to build these integrated cells and to analyze the variation. What we want to do is take these cells and move them through state space. So understand how they change through the cell cycle, through different states like differentiation, and in response to perturbations like specific agonists or mutations. So today I'm speaking in this wonderful session called Maximizing the Value of Your Image Data, and I'm going to tell you one story that relates to two different types of data, one which is one single movie of our cells, and the other which is a very large collection of 200,000 3D images of our cells. So we start with cell culture, we do image acquisition, but we've worked on a lot of methodology to create those data sets using as much standardization and reproducibility as possible. So we use a robotic platform for automated cell culture maintenance and sample preparation. When we're imaging, we use automated methods in order to identify the location within our field of colonies that we want to image, a particular field of view. And our workhorses are generally spinning disk confocals. We also have some semi-super resolution area scan, and we image thousands of replicates of live cells in high resolution and in 3D. We have to handle the storage, image processing, tracking, but we try to do all of that as much as possible with open source software and tools and approaches that we can then also share with the community. So here's what our cells look like. If we move in 3D from the bottom to the top, we can see that the cells are outlined in purple, and in this particular case, we're also labeling mitochondria. And if we look at a visualization on the side, what that looks like in 3D, you can see our cells grow in very tightly packed colonies, and the third dimension matters a lot. These cells are apical basal, epithelial-like. They have a top and a bottom, and they contain very large nuclei. If we take the channels apart, this is what they look like. And of course, we can start and say, okay, we've got our cells, we've got our structure and our DNA. Let's just segment the cells and nuclei, pull out the cells, and let's move on. The problem is, of course, this is all in 3 dimensions and very tightly packed cells, so this is quite hard. And the other challenge is that we're not doing this for one structure, but in this case for 25 different structures, and finding ways to do that successfully, even in a first pass way, is quite challenging. So we created a software toolkit that we use in-house that helped us a lot with this, the Allen Cell Structure Segmenter. It combines some classic best segmentation algorithms to do the structure segmentation, together with deep learning segmentation via iterative deep learning specifically to get to our cells and nuclei, and I'll show you what that looks like. So this is the framework for the Allen Cell Structure Segmenter. It has one classic image segmentation workflow. That's what we used for the structures. And if you look at these particular images and the Z-stacks as we move through our cells and you find this interesting to your own work, please come and check out our tool. We've tried to build it in a way that it's modular for many different uses of many structures within the cell, and just come and check out website out. The second part of our workflow is to use iterative deep learning, where we use the output, which hopefully has been optimized in a way that you like, of the traditional segmentation in order to create training sets to then do deep learning segmentation on. And there's just one curation step needed here. It's very difficult to do a lot of manual curation in 3D. So instead of doing it in 3D, we do it by creating these segmentations and doing either sorting or merging to create those ground truths and then build our models. And that's what we used for our cells and nuclei. There are video tutorials available for both of those workflows, and we have a forum online. So please come check this out and see if it's useful for you. Here is an example of what our cell and nuclear segmentation looks like. So this is the DNA and this is the membrane. This is what the model output looks like, and that's in a whole field of view. So to get to individual cells, we combine the DNA in order to seed watershed for cell identification and the cells in order to refine the DNA. And if we look at this now in 3D, you can see beautiful, beautiful segmentations in 3D of all of our cells. This is a fun little 3D way of looking at those. And when we did a validation by a manual inspection, we found very, very high segmentation accuracy for individual cells and even entire fields of view, which turns out to scale up actually quite well in a fully automated way to our 200,000 plus cells. So we have those tools in hand and we have a way to pull the information out. What do we do with it now? Well, the challenge comes from the fact that cells themselves need to generate robust and adaptable behavior. And the solution that the cells have for this is that they generate structures within, but those structures are stable in a dynamic sense. They are constantly undergoing flux of their components so that they can generate structures that look relatively stable, like these spindles, all while being able to be flexible and responsive to the needs of the cell. In this case, for example, the cell cycle. And these green structures here, those are mitochondria, just to show you that the insides of the cells are constantly in motion. The consequence of that is that if you took any of these cells, this is a field of cells here, and took a snapshot, they would all look completely different at any moment in time. And even through time, they might take different trajectories. So that means we just have this vast amount of variation in our cells, and we have to deal with it. So why do we care about variation? The first reason is because we do have to deal with it. We can't decide what normal is in this complete sea of variation, but we don't understand where it's coming from and how you start creating something that is abnormal. So we need this for a baseline. But we want to go one step further and really take advantage of the variation. And this is one of my favorite quotes by Rick, who is the director of our institute, which is that every cell is an experiment in this sea of cells, because every cell may be slightly bigger or slightly shorter or slightly flatter at any moment in time in what is completely normal. But in a sense, every cell is a slight perturbation on what the other cells are doing. So if we could understand what the sources of variation are, we can then control for it so that we can actually compare cells and identify patterns and rules that generate the organization that we see. So conceptually, that sounds all fine, but how do we actually go about doing that? So here are 4 movies, 4 structures. The cells go in colonies, so these are 4 sort of colonies. And this is just to show you that there's a lot of variation in behavior in the case of the nuclei and the cell boundaries. If we wanted to compare 2 cells, we certainly know already, most of us as biologists, that we want to take into account the cell cycle. A cell that is small and a cell that is large, they're just in 2 different parts of their growth, probably shouldn't be directly compared without taking that into account, because the larger one would have twice as much of all of its internal components. Similarly, again, the behavior of cells in time, whether they all explore the same space within one particular trajectory of time, and you're just catching them in a specific moment, or whether they all have a very particular trajectory that they take, is really important to understanding how we compare those. So we want to be able to take all of these structures, look at how these sources of variation affect what these structures do, and start to put them together. And that's not so straightforward. So let's start really basic, really easy, and see if we can do that conceptually. Let's start with just the nuclei. We all look at this picture, we all start to think, yes, we could analyze the shape and movement and behavior of each one of these nuclei, so let's do it. Well, the first thing we need is robust and accurate segmentation of all these nuclei. That should be no problem. I just showed you our wonderful segmenter and all of our tools. Let's just do that. The problem is that this particular movie, and any movie like it, where the cells can be imaged in 3D for very long periods of time, hours on end, in fluorescence, generally works best for lower resolution and magnification. And if we image our cells at the resolution we would want, here's a side view, to pick up beautifully the shape of these nuclei, then we kill the cells. So we have this trade-off. The question is, can we transform our images? And the answer is yes, we can. Several groups out there, ours included, have approaches for trying to transform images from one type to another. In our case, we do this completely in 3D and with a lot of validation, and we use deep learning conditional GANs, CGANs, to do so. And we call those transfer functions. When we do that and we look at a real high magnification versus a predicted high magnification image, we see very low error in the real versus the predicted, in this case for the volume of the nuclei, which is something we do want to measure. So we do an application-specific quantitative validation in any case that we use this type of a tool in order to facilitate our work. So with that, we can now, in a really beautiful way, and in time lapse, analyze the 3D shape of these nuclei. Oops, apologies for that. So now we still have to track the nuclei. That's a straightforward thing, but not always that easy. So here we are, here are our tracks. And what this basically does is it transforms, let's say, a field of view of a high magnification set of nuclei into a very, very large field of high magnification set of nuclei so that we can actually analyze them. So then we have beautiful 3-dimensional nuclei, and we have to pull numbers out of them to describe shape. We use spherical harmonics to do this. We generate the coefficients that describe, in increasing accuracy, the shape of the nuclei. And then we perform a principal component analysis to identify those primary axes or modes of shape variation. But then we want to understand them. So here we've plotted just cells from the first versus the second component for these nuclei. And here's a view from the top and two side views of these nuclei. And as we hold the second principal component axis fixed and we move through the first, we can see that the nuclear shape is changing. And if we do that more in time, we can see that more actively. We can do that for all of the other principal components. So this very first one that you're seeing here is that basically the size of the nucleus grows. This is a top view. The second principal component would show that the nuclei are squishing. The third would show from the top view that they're getting pressed. And then the fourth and fifth relate to these other sort of versions of getting squished in the other dimensions. So I'm going to summarize a lot of work we did using this approach on this one movie to tell you what our conclusions were. And they're actually pretty fun. So if we look closely at how through time, the mode that presents the squishing of the cells behaves, we can see that how squished a cell is at any moment in time depends on its neighbors and what it's doing and how it's moving, and that happens at the time scale of minutes. If we look at size and we follow any one of these nuclei, you'll see that they grow over time at the scale of the cell cycle time. And importantly, that component, that flatness component, how flat the nuclei are, that relates to how the cells pack within the colony, and that's a much longer time scale. So if we want to ask how much variation is there between two cells in time, it actually depends on the aspect of the variation that you're looking at. And each of these sources of variation map to these three different time scales. So the type of variation, right? Do you care about size or do you care about flatness? And the time scale of a variation affects your interpretation of your data. So that's a good approach. Does that work for our snapshots? Does that work for a huge collection where we have all of our many, many structures? So this is our image collection, which we're updating currently, and hopefully will be updated online in a few months, of 200,000 cells using these segmentations with 25 structures, all high resolution 3D images of live cells, for a collection that we really think is quite unprecedented and powerful if anybody wants to come and play with it, just like we do. So we do the exact same thing. Let's just do the spherical harmonic-based PCA shape space. That's a lot of words, for the cells and nuclei in our human stem cells. And this really is a way to measure and quantify the shape of every one of our cells in the collection in a standardized mapping framework. And if you look here, this is a side view for the very first component. This is a top view for the second component, and so on. The first observation you'll see is that the first 5 modes are the same as in the time-lapse analysis. Some of the order is switched. And in the case of, for example, flatness versus size, that's because in one case, we're focusing on time-lapse of cells through time as they grow. And the other case, we're focusing on many, many still images of cells from many, many colonies that are all in a different sort of space of packing. So that makes a lot of sense. The other thing that's really fun is, besides flatness and size, if you start looking at these other modes, for example, this mode of tiltness that's almost like an Egyptian walk, or one of my favorites down here, which is how pear-shaped the cell is, if you look from the top. You can see that nuclei can change their shape dramatically, and sometimes they don't change their shape at all, and instead they're actually changing their position. So the nuclei tend to follow the shape modes of the cell in some sort of way. So we can now decompose and just briefly talk about each of these modes, just a couple of them. The first is flatness. This in the cell also relates to the packing of the colonies, which is affected by things like colony size, where you are in the colony, and so on. An important one is it could be affected by the tagging of the cell lines. So we did an extensive analysis to look into and make sure that how our colonies pack is not simply due to the fact that we have tagged one particular protein or another. And we found that out of our 25 cell lines, there are a few where they pack a little bit differently from the rest of the population, but not by very much. And for most of the cell lines, which is actually quite spectacular, for all of these 25 structures, for most of them, they actually behave identically. If we look at size, that's growth during the cell cycle. What this permits us to do, of course, is think about, for our many structures, so at least 10 out of these 25 are cytoplasmic structures of interest, how does structure size scale with cell size in a survey manner? What I mean is, how much of the variation we see in the volume of a structure, say the mitochondria, is explained by just simply the volume of that cell. What we find is that for many structures, it's quite well behaved, and there's different amounts of variation that are explained just by cell volume. And for other structures, that's not so much the case. What we also find is we look at structures inside the nucleus, more of the variation is explained when you consider the nuclear volume than the cell volume, and so on. So this is actually really exciting, and I don't have time to show you all of the results, but the idea that we can think about structure size scaling in this sort of, broad way is really wonderful in our data set. All of these other modes, what they're really doing is changing the way that the cytoplasmic space looks and also what the nucleus is doing. And so that means that now we can start asking questions about where all of our structures are in the cell, given a particular sort of shape of the cell. So I'm going to show you a preliminary sneak peek of what that looks like. Preliminary because this is not finalized. Some of our methods are still being adjusted. If we take now the structures, we can ask what are the locations in cytoplasms of similar shape. We can create average locations for these structures in a much more simple way. It's not simple, in a much more straightforward way than if the cells have many different shapes. So we created a very elegant morphing and interpolation approach to start combining cells of similar shapes. So let's take this particular mode here. And here you can see 22 of the 25 structures, and the average view, this is a side view of the cell, of each of these structures. The pexillin, which is for the focal adhesions, is at the bottom of the cell. Tight junctions is near the top of the cell. They're beautiful patterns for some of these organelles. But what we can do now is we can look at how these patterns for each of these structures changes in response to natural perturbation. For example, when the cells are more flat or larger, or in this case, sort of more elongated in one way or the other. We can also start, and this is also preliminary, but very exciting to me, looking at the locations of all these structures relative to each other. How much overlap is there? So we can take the same particular shape space and we can start painting the structures in. And I can't even overlay all of these into one because it would look like a big mess. And so we can visualize here 13 structures in the same integrated cell, and we can do that through changing the shape space. And then we can try to think about how to represent that. So this is, again, very preliminary because the particular approaches we're using to measure the overlap are still being optimized. But if you take each node to be a particular structure that is represented by some protein that is tagged in our cell line, and you take the strength of the connections to be sort of the thickness and darkness of these lines between the nodes, then what you'll notice is these set of structures here are all nuclear. And they have strong overlap with each other, as you would expect. They have less, but still some overlap with the nuclear envelope structures, which all overlap a lot with the endoplasmic reticulum that forms a bridge to the cytoplasm. And then there are structures over here, which represent structures that are towards the top of the cell or the edge of the cell that then interact with the structures at the periphery of the cell. So this is a mechanism we have to now create structure interaction maps in our cells, and that's exciting and fun, and we can see how it reacts to whether the cells are bigger or smaller. But we want to take that one step further and say, now, how does that change when cells change, right? When we go through this differentiation process. So just briefly in the last two slides, just to tell you where this is heading. If we now look at the early time points in differentiation when the cells transform from epithelial-like cells into mesenchymal-like cells as they form mesoderm, and they go through an EMT, which is something that occurs in differentiation, but also in cancer and inflammation. This is what our colonies look like. So individual cells in the colonies are labeled. What you see is that the colonies contract very dramatically. And then at about 24 hours, they expand and the cells start moving around and crawling around with a completely different morphology and organization than before. And if we look closely at the nuclei, one of our wonderful prototypes for analyzing this, we can see all sorts of important changes in how the nuclei behave in shape space. And so analysis of these sorts of trajectories will allow us to get at whether what types of behaviors occur during really key state transitions like developmental differentiation. So with that, I want to thank the whole Allen Institute for Cell Science team. We have extensive contributions from everyone on the team. We all work together very closely. And I would like to end by thanking our founder, Paul Allen, for his vision, encouragement, and support. Thank you.
Are we back? Okay, thank you. Sorry, guys. Thank you, Susanne, for this very, very interesting talk. So we have several questions from the audience, so let me go through them. First question was, how do you define the edges between colonies that are touching?
Yeah, so, oh gosh, you guys can't hear me yet. Can you hear me?
I hear you.
Good, here we go. Great, let me just add my video. Hi, yeah, so we can't necessarily define edges of colonies that are touching. So if we want to do colony analysis, we would focus on colonies that are separate from each other.
Okay, another one. Well, first, complimentary. This is incredibly cool. For the shape modes, are there any indications that some of the shape modes change in correlated ways? For example, pear-shaped cells tend to be smaller, etc. Have you tried any non-orthogonal dimension reduction techniques to see if some of them provide the same account of the data as PCA?
I love that question. We have looked at whether some shape modes are correlated. Some of them are, but not in a huge way that affects the results. However, we have a parallel approach that we're trying to use to actually compare different methods of doing dimensional reduction. We're doing this right now with PCA, but we're also doing this with a beta VAE autoencoder approach, or a machine learning approach, because that would be nonlinear, and it would allow us to see whether the cells want to tell us something different about how most effectively biological information should be compressed and then expanded again. So we're actually going to compare both approaches, but the beta VAE approach was not yet ready for sharing. So.
Next question. Okay, another question, also a compliment, great talk. And the question is, can the shape mode also be used to quantify nucleus in time-lapse of bright field images in 3D?
So yes, with some work. So we have a label-free technology where we can predict the fluorescence signal of the outline of a nucleus, or potentially even segment directly the nucleus from bright field. And we're working on that, and others can work on that too. And if you extract the nuclear shape properly from bright field via these label-free approaches, you could absolutely do this. And we are trying very hard to get there. So.
Okay, another question. So every question starts with a compliment, so I will not repeat them. But one question is asking, what is the relevance of where the structures are in a cell? That's a good question.
Yeah, so our goal here is to say, if we look at an individual image, we know where something is in the cell. But do we know the rules of which structures localize where in relation to each other in a consistent way and in response to the perturbation, for example, larger or flatter cells? And so the relevance of this is sort of that point, which is, what are those rules by which structures localize with each other? Are those rules consistent over perturbation? And how do they change when we go beyond, say, shape space for these cells, but say differentiate into another cell type? Are the Golgi always closely related to the lysosomes in only our stem cells, but also in the same way in, say, a crawling cell? Right? Those are the really core questions of rules of organization we're trying to get at.
Okay, I think we have to wrap it up in the interest of time. Thank you, Susanne, for a very interesting and stimulating talk. And thank you, everybody, for listening. If anybody has their question not answered, I think Susanne would have a chance to later answer your questions in writing, I think. So that's it. Thank you very much again, and enjoy the rest of the conference.
Thank you.